Morphology and structure of the virus. So in this chapter we'll be studying three things. One is concept of virus here, and then another is morphology, and another is structure and chemical composition of virus. So the first one, concept of virus, there are different terms we need to know. For example, virus, virion, viroids, virusoid, prions. So what are all these? Let's discuss that first. So viruses means those which contain DNA or RNA, and then it is encoded by a capsid. Capsid means the outer layer, and then it may or may not have outer lipid membrane called envelope, and then it infect all the organism that are virus. That is virus. Then what is virion? Virion are those which has inside DNA or RNA capsid, and they also have envelope in the outer membrane, like this envelope. They also have envelope, and in the envelope there is this spiky things called spikes of protein molecules. So basically what we can say is virus can be divided into two parts. One is naked virus, one is envelope virus. Naked virus is the one without envelope and this protein molecule at the back, whereas envelope virus, also called virions, are the one which have a capsid and envelope and this protein membranes. Okay, and there is another thing called viroid. What are viroid? Viroid are smaller than virus, and they have a RNA inside, and then they can only infect plants. Then those are viroids. Now what about virusoid? Virusoid, I think it is also called satellite virus, something like that. It possesses linear or circular DNA. Linear means straight, circular means circle. RNA, and then it can only infect what? How does it infect? It needs a helper virus, and then it will after helper virus it will go in the host cell, and with the help of that it will replicate into the new virusoid. Now what is prion? Prions are those protein which is like, at first there is some normal proteins encoded by the host chromosome. So in the host chromosome there is a normal protein which will change into misfolded protein, infected protein, which can cause disease. These are called prions. Prion, I think it's pronounced as prion. Okay, that's all. And the size is like 20 to 300 nanometer. So basic structure of viruses, it's acellular. Acellular is single cell. Obligate intracellular parasite, which means what does obligate intracellular parasite means? Let's see. It means that cannot be produced outside their host cell. That means it is entirely dependent on intracellular resources. That means in the host cell, the resources present in the host cell, it is dependent on that. Okay, then there is no ribosome, there is no ATP generating system, there is no mitochondria. So, and then they have either DNA or RNA, but not both. And morphology, there are different types. One is like like a ball, spherical shape, brick shape, and then filamentous virus, tadpole shape, bullet shape, Singan pinto it's like spherical shape maybe. Then size. On the size, normally we already read here size is 220 to 300 mm, and nm is nanometer. So smallest one, what is smallest one? It's paravirus, smallest one. Big one is Tianhua Pingdu. This is virally virus, it's also called virally virus. Okay, so how is this Pingdu T the Jego? What is the Jego looks like? Let's see. It looks like this. Let's understand inside this. Okay, so there is inside nuclear capsid. It consists of two parts, core and capsid. In the core there is DNA and RNA. Then there is capsid, which is made up of capsomere. We'll learn that. And then outside part is called matrix protein, and outside that there is envelope, and outside that there is spike. Spike is called SIR2. So I told already there is a virus. Virus is two types. Then that is naked virus and envelope. Naked virus means they don't have envelope and spike, whereas if it contains envelope, palmo, and then spike, I think it's called, then it will be virions, ping to T. So naked virus without the envelope, enveloped virus with the envelope on protein. Example is influenza A virus, rabies virus. Okay. Then we have to learn something about Epstein-Barr virus. What is this Epstein-Barr virus? This virus is, let's see, what is this called? This virus is also called monovirus or kissing disease. It's not kiss or what? Okay, so it has symptoms of fatigue, fever, and this kind of thing. Epstein-Barr virus. This is what it is. And then, what does it infect? It normally infect accompanied by a large typical large peripheral blood lymphocytes, meaning the lymphocyte, which is, what is lymphocyte? Lymphocyte is a type of WBC, white blood cell, which works for our immunity. It affects that lymphocyte, causing what? Causing carcinoma, lymphoma. It causes those kind of things. It affects P and I. I don't know what is this. Nasopharyngeal cancer, something like that. Yeah, that is what it causes. Okay, after this, next one. That's the next one. So subviral agent. So what is subviral agent? It's like there is a big something is unit, and then inside if it contains different part, that is called subunit, something like that. So virus is like a main part, and then inside that is sub. Those are like viroid is one part, satellite virus, the one I said last time, and then prions. We already discussed all this, right? Viroid being it is made up of single stranded RNA, and then what else? Single stranded RNA, right? And all the things we learn. Then it is only present in this plants. Then satellite is like virusoid, which is also in plant. It helps in helper virus. Then prions are the one prions are the one which cause lesions, mad cow disease, something like that. So yeah, these are also done. So next, viral genome. First of all, I need to know what is my genome, right? Let's see in another page. Yeah, satellite, that I already told. What is that? Satellite RNAs. Then genome is like, what is genome? Genome is a complete set of genes present in a cell. Complete set of all the genes present in the cell are called genome. So what is the characteristics? So in virus there is either DNA or RNA, and then diversity is like single stranded, double stranded, round, circular. And then another point is economical. That means it will use less possible gene to with a more multifunction, that is called economical, like a minimalism kind of thing. Then there is replication mode, and then there is it is classified on the basis of first one, that is DNA or RNA or its sequence, different kind. It is classified. So now let's see the classification. For example, it can be classified on the basis of what is present, DNA or RNA, or on the basis of what kind of DNA or RNA is, a double stranded, single stranded, different thing. For example, double stranded circular gap, single stranded or linear is the straight one, linear gap, single stranded linear. In the RNA also there is something like that. So it can be classified on the basis of linear, circular, and then single stranded or double stranded, or monopartite or multipartite. Monopartite means continuous without any gap, whereas multipartite means segmented, like gap here. Yeah. Okay, so now we learned something about, let's learn about single stranded RNA, ssRNA. Single stranded RNA, let's learn. Okay, so what is the single stranded RNA? It is also called Tanzanglian RNA. Let's see what is Tanzanglian RNA means. So So positive strand RNA are a group of related viruses that have positive single-stranded genome made up of ribonucleic acid. What is this? It means simple. What we need to understand is positive sense genome. This kind of RNA can act as a messenger RNA, and it can directly translate it into the viral protein. It can directly translate into protein. That's what we need to know. That's what it is written here. RNA can be translated in acts as mRNA and translate the protein. Whereas if it is negative single-stranded RNA, what will happen? This negative single-stranded RNA will first produce positive single-stranded RNA, by a complementary base pairing, something like that, and then it will act as an mRNA and then translate the viral protein. Yeah, that's it. Next one. So vertebrate DNA viruses, different names of the viruses we need to know. Okay, and then which is present in vertebrate, and then different RNA viruses present in vertebrate. That's it. So the size is like, what is size? Size is measured in terms of BP. What is BP? BP is base pair. Let's see the size. Okay, size is a base pair, which is like 1 kilo basis is equals to 1000 base pair, or 1 MB equals to how many is this? 1, 10, 100,000, 10,000, 1,000, million, 1 million base pair. What is my this base pair now? Do we know what is my base pair? Base pair is a fundamental double-stranded nucleic acid consisting of 2 nucleobases, meaning these blue things 1 and 2, these 2 will make a base pair in a DNA is double-stranded, right? Yeah. So in one DNA, like let's just say how it is calculated is 1 base pair is equal to let's just say 10 of the base pair, this side is pyrimidine, those kind of thing. 10, 1, 2, 3, 10 base pair is present in one complete helical structure of DNA. Yeah, 10 is present. So like that, how many is present? 3200 is present in hepatitis B, likewise. Okay. Next one is, do you remember in the previous property we learned like economical, this viral genome is economical, meaning, for example, same sequence can have like different function. For example, when 2 of the protein is overlapped, then it can function as it can work as a compilation area and regulation area, both. That is called economical. This is an SBB virus. Okay, now let's learn different parts of the virus. What is capsid? Capsid is this capsid is made up of capsomere. Now let's know what is capsomere. Capsomere is a subunit of a capsid. So small capsomere, when it comes together, then it will make capsid. Yeah, capsomere self-assemble to form a capsid. And then how capsomere is formed? Capsomere is formed like this. Capsomere is composed of different types of protein, and it will repeat over and over and over again. Small repeat protein, and then it will make whole entire capsid, which covers nucleic acid. That is what capsid is. How is capsid formation? We already talked about it. It keeps on repeating, and that's how it forms. There are different types of structure. For example, isohedral symmetry, then helical symmetry, complex symmetry. So this, what is the function or uses of this capsid is, it can work as a protection of nucleic acid in the inside, and it can be a sites of infection, or it acts as an antigenicity, or it can be used as a classification, like this type of bacteria, this type of virus, right? What is this antigenicity? Means, let's see, this means antigenicity is a capacity of a chemical structure. What is structure? Capsid. Capacity of the capsid to bind with a certain group of certain product. That means antigenicity means in a virus, capsid will bind with some product that have an adaptive immunity. For example, T cell receptor in our body, like there is T cell, right? So it's basically a capacity of a structure to bind, to combine. Okay. Next, these are also different kind of types. So now let's learn about envelope. Envelope power. What is envelope? Remember that structure here of the virus at the back. Here envelope. So this outside is capsid, right? So this one is envelope, and inside this is protein, matrix protein. Let's see what this envelope look like now. Viral envelope is what is viral envelope made up of? Viral envelope is like, how does it works? So this is inside the host cell. When it is inside the host cell, how does it work? No, let's not talk about how does it works. Let's say it is made up of what? Double layer lipoprotein, and then there is a spike at the end. This spike. So spike is found in a host cell first, yeah, and then this will cover the virus and it will get out of the cell. So this is how envelope, and then spike comes, yeah. And then properties of this envelope is what? Sensitive to fat, and then if naked virus is there, that is, it doesn't have spike and envelope, then it is more sensitive to heat. Yeah, that's it. It can be also, the viruses can be classified on the basis whether they have this envelope or not. Okay. Now matrix protein. Matrix protein is the one which was inside the envelope. So this is envelope, this is spike. Inside it, here is matrix protein. It is also called tegument. Matrix protein is also called tegument, divided into inner tegument and outer tegument. Yeah. So what to learn about on this? Nothing. It is present in some example is given, some virus example like paramyxovirus, retrovirus. That's it. Let's say about what now? Let's talk about different types of protein present. It is divided into like enzymes and non-structural protein. Enzymes, what are the enzymes present? It is produced inside the body of the viruses. These enzymes are DNA polymerase, reverse transcriptase, protease, neuraminidase. Whereas non-structural protein are those protein which is what does it do? It for example, it helps in inhibition of biosynthesis or regulate immunity. For example, for this HBV virus, the non-structural protein is HBAG. What does that mean? It means, see, for example, HBAG is a hepatitis B viral protein. What does it do? It means person infected with hepatitis B can likely transmit virus to another person. Meaning what? Meaning if we found this HBeAg, hepatitis B antigen, we will find this in the blood. So that is how we can test whether the person got this hepatitis or not. That's how we can check. These are non-structural protein. In this HCV virus, see the genome, half of a little bit is structural and other is non-structural protein. Yeah. Now something about coronavirus. It is single-stranded plus single-stranded RNA. Remember that positive one which can act as a mRNA, messenger RNA. It is coronavirus. Then the size is here, and then this is kb, something kilo base pair. So it is base pair. Kilo is around 1000 base pair, right? So around 27,000 base pairs are present in COVID. So what is ICTV? ICTV is International Committee for Taxonomy of Viruses. Taxonomy, that means it classify the virus on the basis of type, sequence, length. What else? You may if it has envelope or not. Then size, replication process, mode of transmission. Different things are there. How is this time's novel coronavirus is named by ICPT? It's novel coronavirus. Then SARS-CoV, it named as novel this NCP COVID-19. Yeah. So this is Xiaojie. What is there? Naked virus and enveloped virus. Remember, virus is divided into two parts. Let's not say two parts, but something like that. Whether it has enveloped or not. On the basis of that, ICPT has divided and classified it. So yeah, we learned about virus, we learned about virion, naked virus, envelope virus, then capsid, and then lots of capsid. Capsomere made capsid, nucleocapsid. Spike is the outermost layer of the envelope. Viral genome gene. Then after that, what protein? Protein is structural and non-structural protein. So viral agent, those viroids, virusoid prions, viroids, virusoid prions. Yeah, that's it. This was for today. Bye bye. Thank you.